of our discussion on protein formulation and delivery. We've already gone through part one on Monday, and I gave you guys a bit of a backgrounder on proteins and protein structure and why proteins are both interesting from a formulation perspective, but also important in terms of the drugs of the future. And so because of their importance, I think we can expect that protein formulation will become more and more important, and also its delivery. Okay, so we'll maybe just dive in right now. Again, if there are questions you have, you can probably put them in the chat. If anyone's having some difficulty hearing me, maybe just connect to the student representative so they can stop and make sure that my video is coming in clear. So as I said before, we talked about protein structure. We looked at different types of proteins, and not quite sure why this is showing up. Different folding classes, and the tertiary structure of proteins, which is basically held together through non-covalent hydrogen bonding or hydrophobic interactions. Proteins, when they do fold into these tertiary structures or three-dimensional structures, either have helices or beta strands or a mix of both, and they fall into these general classes. And with structure comes function, and it's a function that we typically are trying to elaborate when we produce protein pharmaceuticals. I talked a little bit about the different classes of protein drugs or protein pharmaceuticals, vaccines. Of course, everyone's thinking about vaccines now with COVID. Some of the vaccines are RNA-based. These are new concepts, but in the end, what the vaccine ultimately is a protein, a fragment of the protein of a bacterium or virus. There are peptides and blood products, and then the vast majority of produced protein pharmaceuticals are recombinant proteins, and we talked about some of them. I also went into sort of an in-depth discussion about the problem of proteins, both in the bottle or in vitro, as well as the problems in the body or in vivo. And I highlighted some of the issues with interactions that change protein function, structure, or stability, and then covalent events that can change or disrupt the structure and stability of proteins as well. So again, this is just a review. And then we went into a more detailed discussion about protein pharmaceutics, and we talked about storage approaches and things like refrigeration, adding different salts. Today we're going to be focused primarily on formulation and delivery, and a good chunk of today's lecture is going to be about protein formulation. We talked a little bit about formulation and site-directed mutagenesis. And we talked about how formulation was really targeted at not so much about storing it, but how to get it in the body, how to make it stable in the body, and how to fend the protein off from various attacks that the body delivers to protein pharmaceuticals. So they're attacked by B cells and T cells. They're attacked by proteases. They're eliminated by the reticular endothelial system, or RES system. And that they all also cause allergic reactions, and they can also be lost through depot effects when they're injected or otherwise taken in. So in the protein formulation side of things, we talked about site-directed mutagenesis, how that is actually the most common way of formulating proteins to enhance their solubility and stability and lifetime in the body. We talked about pegylation as well, the use of polyethylene glycol, which is used in a number of protein products now. We talked about proteinylation, where you're sticking on soluble protein like serum albumin or antibodies. That's still sort of at the experimental stage, not widely used. And then today we're going to talk about microspheres and nanospheres as ways of encapsulating proteins. And we'll also talk about permeabilizers as another route to protein formulation. So microspheres are spheres that are measured in the micron size, typically 100, 200 microns. And microspheres can be made of any number of materials, and they can be used and have been used to deliver small molecule drugs, but there's also work to try and develop them to deliver protein drugs. And we'll talk about some examples. So with microspheres, you're encapsulating. That means that the protein or peptide is in generally a porous particle. And that the porous particle is really there to protect the protein or peptide from the B cells, the T cells, the proteases, and so on that we talked about. The particles not only protect, they also help with the release. And proteins or peptides can be released from these microspheres through a number of different processes that we'll talk about. Now, microspheres can be two types. There are ones that are biodegradable. These are things that can be chewed up by the body or which naturally erode or fall apart. And then the non-biodegradable ones, which are basically more like ceramic microspheres, things that will never break down. They may pass through the body and be eliminated, but they won't break down. So the non-biodegradable microspheres that have been looked at, explored, are ceramic particles, clay particles if you want. Others are made out of polyethylene co-vinyl acetate. Another one is through polymethacrylic acid and polyethylene glycol. So these are plastics basically. So they're either clay or plastic. And those are the non-biodegradable ones. The biodegradable ones, which are generally preferred, are things like gelatin or jello, and polylactic co-glycolic acid, or PLGA. The non-biodegradable ones tend to be somewhat hydrophobic, whereas the biodegradable ones tend to be hydrophilic. And for most proteins and peptides, you have to use relatively hydrophilic things, whereas for small molecule drugs, it's okay to work with hydrophobic carriers or microspheres. So this is on the right side some electron micrographs of PLGA polymers. You can have polyglycolic acid and you can have polylactic acid, but when you mix the two together, you get this polylactic co-glycolic acid, and it can form into various strings or plates, and that's characteristic of many polymers. But it is a plastic-like substance, which is very biodegradable. The ester links are very easily cleaved in water, but also by esterases in the body. So when we look at microspheres, the very hydrophilic gelatin ones are often done for essentially burst release. The molecules come out very quickly. And that's again because they're so hydrophilic. Water easily permeates, they are very porous, and both small molecules and proteins can be removed from gelatin capsules very quickly. PLGA is somewhat hydrophobic. It's not fully hydrophobic, but it is something that can be used for sustained release. And that's a characteristic of hydrophobic microparticles in general. Now, for large proteins, PLGA isn't the best because the proteins tend to denature in PLGA. But peptides, like oxytocin or pitocin or smaller molecules, even ones that could be used for vaccines, can be put into PLGA. Now, what you want in the end is something that combines both, the gelatins that can solubilize proteins and PLGA, which can release things slowly. So these amphipathic or hybrid, hydrophobic, hydrophilic polymers potentially offer the best route to sustained release, but as well as keeping things native. Now, lots of work is going on to developing these amphipathic polymers, but nothing yet has really made it into the market. When we've looked at the different routes of how microspheres release their payload, whether it's small molecules or big molecules like peptides and proteins, there's natural diffusion. So something that's porous, stuff will just diffuse through those pores. There can be degradation, and that's true particularly of biodegradable polymers. So these things will break up. They'll be either hydrolyzed if they're esters, or they'll go through enzymatic degradation if they're amides or peptide or ester forms. Obviously, the biodegradable polymers or microspheres will both experience standard diffusion as well as polymer degradation. And then hydrolysis, which is just the wearing down from exposure to water. You can have erosion, which again is just simply things are chemically and mechanically breaking down. So that's another release mechanism. So these are all the things that we're sort of exploiting within the body. The fact that the body has enzymes, it has water, that it has flow characteristics that can be used to erode or diffuse or break down the plastics or the polymers in these microspheres. Now, another approach to delivering peptides and proteins is to use things. And you guys have probably heard about liposomes and micelles for delivery of small molecule drugs. So these are, made up of lipids, and they're very, very hydrophobic. Unfortunately, you can't really do lipid-based delivery of proteins because, again, that'll denature the protein. So the approach that people have been thinking about and looking at for a while is, why not make micelles that are more protein-like? And so rather than using lipids, why not put essentially a peptide-like or protein-like core to the micelle? And so that's the name peptide micelle. Now, the micelles have advantages over, say, larger liposomes in that they can be very small. And when you get things that are on the order of 10 to 50 nanometers, they evade a lot of things that the body has for defense, which is why viruses like coronavirus can be so infective. So if you make something small, then it avoids some of the immune response. It has a lot of the behavior of lipid micelles. But as I said, with peptide micelles, we don't want lipids because that denatures proteins. We want proteins or peptides, and peptides like peptides. They dissolve like dissolves like. So we can make the peptides mildly hydrophobic, and then we can cover those peptides with polyethylene glycol. And recall how we talked about PEG, or polyethylene glycol, is very hydrophilic, very biocompatible, and that forms the surface of the micelle. So as I said, the peptide core gives you a chance to dissolve proteins nicely. You can also put in small molecules into peptide micelles as well. To make a peptide micelle, you have to make a monomer first. The monomer has to be made of a peptide, and so you can use peptide synthesis to construct this 10, 15 residue peptide. And you can design the sequence in any way you want. Peptide synthesis has been around since the 1960s. There are automatic machines that do this. My lab used to do a fair bit of peptide synthesis. And this is how the synthesis process is done. It's very efficient. You can make peptides up to 100 residues long on a peptide synthesizer. After you've made the peptide, then you can couple, using the same machinery and chemistry, polyethylene glycol, or PEG, to the peptide. And so the entire thing, the monomer, if you want, is made on a solid phase synthesizer off the peptide and the PEG from the synthesis beads. And now you have, billions and billions, trillions of monomers that you can dissolve in water. The peptide is, as I say, relatively hydrophobic. The polyethylene glycol is very hydrophilic. And so you'll get a spontaneous event of micelle formation. So this is just showing, I guess, in a cartoon, if you want. You can see the monomer, which is the yellow peptide, and the polyethylene glycol solubilization component. And then you can see some proteins floating around in solution. And the monomers will start self-assembling, and they'll start forming, in some cases, around the protein or polypeptide, or they'll just spontaneously assemble themselves. And so the yellow core surrounds the protein, and then this hairball of polyethylene glycol starts forming. And this hairball here is essentially a fully formed micelle with a protein or peptide or several proteins engulfed by this peptide polymer monomer to form the micelle. So that's how peptide micelles form. It's how they can encapsulate proteins. You can also play around with modifying the surface of that hairball and put on targeting molecules. And that way the targeting molecules can bind to receptors, and so you can make the micelle just like a virus. It binds to a surface, and then it's engulfed by the cell and releases its payload. Now, micelles are examples of nanoparticles, and I talked about the microparticles. You can also make nanoparticles out of other derivatives, certain types of plastic and silica and carbon nanospheres. And those nanoparticles can also be used to deliver proteins. Now, again, many proteins are not stable in nanoparticles, and so generally vaccines are better, or short peptides. If you can get those nanoparticles to go into dendritic cells, then you can actually make some very powerful vaccines. So dendritic cells are essentially immune cells, and they go around eating pathogens and presenting the antigens to T cells and even to B cells. And so they secrete cytokines to help manage the immune response. So they're really critical and important cells for creating powerful vaccines. So if you can get dendritic cells to eat up peptide vaccines, then in fact you can make a very useful vaccine. So people are exploring the use of the nanoparticles to get peptide components into the dendritic cells, and they've been using these for both viruses like AIDS and hepatitis B and C viruses, but also developing cancer vaccines. This is a picture of some polymer nanoparticles, pseudoplastic ones that had actually been taken up by dendritic cells. And so the payload, the protein, the peptide that they have in there can essentially be converted and presented on the surface of the dendritic cell and induce the T cell response. So vaccine delivery is another route and where essentially nanoparticles are being used now. So in addition to nanoparticles and microparticles to get proteins and peptide vaccines in the right places, people can also add other components to a protein formulation. And one thing that you're always challenged with is, how do you get the protein from point A into the body? How do you get it from the skin into the blood vessels? How do you get it in from the mouth to the blood vessels? How do you get it from the stomach into the blood vessels? How do you get it from the small intestine into the blood? And one way of doing that is literally by punching holes in the surface. And many of you know that aspirin is a permeabilizer. It punches holes in the gut, and people that take too much aspirin will have bleeding and can develop serious ulcers. So by adding essentially salicylic acid or aspirin derivatives, you can actually induce small micropores that allow proteins to actually permeate from the surface or a stomach lining or an intestinal lining into the cell or into the bloodstream. Fatty acids also do the same. EDTA, which is a metal chelator, also can do the same. And there are other things that people have found that can induce holes or pores in tissues or in barriers, even the blood-brain barrier, to get small molecule and big molecule drugs across. So this is another formulation trick. It's obviously different than microspheres and nanospheres, and different than site-directed immunogenesis, but it is considered a formulation approach. So just to wrap up, last time we talked about protein sequence modifications to help with protein formulation, site-directed immunogenesis. So we talked about pegylation and proteinylation. Today, we talked about microspheres and nanospheres, ways of encapsulating peptides and proteins, both for vaccines and for protein drugs. And then we've also talked about permeabilizers as ways of allowing proteins to get into the bloodstream or the site that you want to attack. So that kind of wraps up our discussion on protein formulation. So we've talked about storage, we've talked about formulation, and now the focus is going to be on protein delivery. So this is the same picture that we showed, I think, last time on Monday, and it's just highlighting the challenges that we have to deliver proteins, either through the mouth. So generally, if we swallow proteins, they're digested. Likewise, if we can get proteins into the bloodstream, they're often taken up by the liver or eliminated through the kidneys. If we try and inject proteins, which is the main route by which proteins are delivered, there are issues. Intravenous injection leads to very high levels of a protein over short periods of time. Subcutaneous injections lead to a depot effect where proteins precipitate, and so we lose a lot of the proteins. If proteins are insoluble, you can develop phlebitis. That's sort of an inflammatory condition at the site of injection. And then we have the immune response and the protease and peptide response, which will chew up any proteins that are circulating in the bloodstream. So there's all kinds of barriers that essentially prevent easy delivery of proteins. And so we'll talk about those in particular. The standard route for about 99% of all protein drugs is through injection or parenteral infusion of some kind. So that could be either by IV or needle. There are other routes to deliver proteins. I'll talk about some of them, which are either oral or nasal delivery. It's possible for some small peptides that they can be swallowed as pills, but you can also do essentially aerosol delivery. There's work at trying to develop patch or transdermal routes. So nicotine patches is being one. There are other approaches for birth control, again, patch drug delivery, but those are for small molecules. I'll talk about patch ideas that people are exploring for protein delivery. And it is possible to get proteins delivered wherever there's essentially a thin cellular layer, 1 or 2 cells thick, which can allow proteins to pass through somewhat more easily. So lung is one route, which is sort of these nasal delivery approaches. Rectal and vaginal deliveries are also possible for proteins, and also through the eye. But those are not widely done and not commonly experimented with. So the vast majority of protein drugs still come in through a needle. You can have an intravenous needle, you can have an intramuscular needle, you can have subcutaneous, you can have intradermal. There are different deliveries depending on either the site that the protein is supposed to act on, how it's supposed to be delivered, where it's supposed to go. And that's usually worked out through both the drug trials and drug efficacy. Again, it depends on the protein, depends on its target, depends on the stabilities, depends on its role. Is it a therapeutic? Is it a vaccine? Those delivery decisions are largely determined by the manufacturer. So if something is delivered by needle, certainly you can get good absorption very quickly. It allows much smaller doses because all of it is essentially presumably getting into the body. It avoids this first pass metabolism of an oral drug. So oral drugs have to go through the stomach, through the intestine, they have to go through the liver and the kidneys, and they're processed. And so if you can avoid essentially the processing by the gut and the stomach, then that ensures that the protein is reasonably intact. It also avoids high or low pH or high acidic conditions in the stomach that is very unfriendly to proteins. But repeated injections or intravenous injections have issues. You can have overdosing, you can have tissue necrosis where you're injecting in the same area. You can get local immune reactions, phlebitis, hypersensitivity. That's also a problem with protein injections in IV. And the other issue is that no one likes getting needles. They hurt. And so if there are better ways of delivering proteins into the bloodstream without hurting, or getting it through the mouth, which is how most other drugs are delivered, is something that people are really, really interested in. So in terms of oral delivery, it's been explored for a long time, as far back as 1922, actually. And one of the first things people realized is that when you try and give protein drugs to people by mouth, they're actually chewed up by the enzymes in the mouth and in the stomach and even in the intestine. We've talked about some of the formulation tricks that people have developed with permeabilizers and with microspheres. So that's one way of addressing that. You still have a pH issue. So if something drops to below pH 5 or 6, many proteins start denaturing. So in the stomach, pH is around 2 or less, and so almost all proteins are denatured. Again, you can address that by certain types of formulations. And then if you can get past the stomach and the acid, how do you get the drug, the peptide, or the protein past the intestinal barrier, which is a very mucus-rich, relatively thick layer of cells, which still is a challenge for oral delivery of proteins. So people have been studying intestinal transport for a long time because it's relevant not only to small molecule drugs, but to big molecule drugs. And there are different types of transport. There's transcellular transport, which takes things from outside the cell or out in the intestine into the bloodstream. There's between cell or paracellular transport, because cells can also allow things to, not only are they forming a tissue layer, but they have, I guess if you want, stitching between them. There's carrier-mediated transport, which requires that the drug or protein has to have a receptor. And then the big challenge, of course, is getting the drugs past the mucus barrier, which is generally quite thick in the intestine and one where many drugs die. So in terms of oral delivery, people have been looking at oral delivery of insulin for about 100 years because it is still the #1 selling protein drug. So people tried. In 1922, they didn't succeed, but over the last 20 years, there have been a few successes, and I'll talk about 3 of them. One is called Exubera, which is inhaled insulin. So it's just like a puffer for someone with asthma. And Pfizer was the one that developed it. And it actually worked really quite well. It worked just as well as injected insulin. And because people didn't have to deal with injections, Exubera was popular among older people, adults particularly. But what they did find is that people who used Exubera developed a cough. And it's not clear exactly why. And because of those slightly negative responses, actually Exubera was pulled. So Exubera existed, but is no longer a product. There's another oral version of insulin called Oralin, with L-I-N or L-Y-N. A pharmaceutical company called Generics produces it. It's been approved in Ecuador and India and perhaps a couple other countries. Even though it's made in Canada, it still hasn't been approved here as far as I know. It takes advantage of the fact that proteins can be absorbed through very thin layers of tissue or cells in the mouth and throat. And what they've done is largely put in stabilizers and additives that prevent the protein from being denatured when it's aerosol, when it's converted to an aerosol. Most proteins, when they are put into fine particles, will denature. But by adding certain stabilizing agents that are a secret, these things stabilize the insulin protein, and it's able to pass through the barrier, blood or tissue barrier. Another company called Biosante has produced something called bio-oral insulin. And in this case, they're using nanoparticles, calcium phosphate nanoparticles. They're also formulating in polyethylene glycol, which we've talked about before and helps stabilize proteins. And they're using milk protein, so something that's very edible, to assist with that formulation and enhance the protein stability of insulin. So this too also appears to have oral capabilities and is going through the approval process. So as I said, insulin's been the one that's been most frequently attempted in terms of getting oral delivery. People have also started exploring smart microspheres. So we talked about the microspheres made out of ceramic or PLGA, but you can also make microspheres that are pH sensitive using the appropriate combination of plastics. And these can work for both small molecules and large molecules. So a microsphere that is closed up and non-porous at pH 2 is good because the payload is protected from the acid that you'll find in the stomach. As the microsphere moves from the stomach into the small intestine, the pH shifts from pH 2 to about pH 7. And then if you can make the material smart so that it becomes more expansive or vacuous, or it expands, then that increases the porosity and allows the payload to be released. So in that regard, the material is called smart materials because it's pH sensitive. So using polymethacrylic acid and polyethylene glycol, people have made these sort of hybrid hydrophobic, hydrophilic, pH sensitive microspheres. So at pH 2, the microspheres shrink, pores are near zero, molecules can't escape. At neutral pH, the pores swell and things can start being released. Another approach that people have tried for delivering proteins very specifically to the small intestine is through this gastrointestinal mucoadhesive patch system, or GI maps. So these look like capsules, more like horse pills. They're fairly large, and they are designed to basically stick to the intestinal wall, the small intestine, and release their payload. So you've got essentially a 4-layer delivery system. One is sort of I guess if you want, a paper plastic backing, which is on one side of the pill. You have a drug-containing system, which has cellulose or methylcellulose and citric acid, which is how many drugs are formulated. But you can also put in not only small molecules, but proteins. And then you have this mucoadhesive glue, which is the polymethacrylic or polyacrylic acid and PEG, not unlike the pH-sensitive material that I talked about. And then you have this pH-sensitive surface layer made of these brand name sort of glues. So the GI maps is illustrated here. And so the plastic coating is supposed to appear on the outside. The yellow material on the inside is the drug container. And then everything on the right side here is essentially designed to bind to the enterocytes in the intestinal wall. So this is the glue. And because you're now at a different pH, the porosity of this mucoadhesive layer and the pH-sensitive film opens up and allows the drugs, small molecule, peptide, protein, to pass through into the cells, and they're being transported through either paracellular, carrier-mediated, or intracellular transport. And so that moves the drug from the outside of the intestinal wall into the capillaries that line the GI. So this is basically saying in words what I was describing with that picture. And as I say, there's these layers to prevent the attack from proteases, the drug-containing layer, which has dispersions and microspheres and liquids containing small or medium-sized peptides or proteins. And then this gel-forming layer and pH-sensitive layer that essentially allows it to stick to the intestine. Now, that's for trying to get proteins in through either the stomach, the intestine, or through the mouth. You can also try and get proteins delivered through the skin. And so the idea is to use these patches. But it's not a patch system that's done for nicotine or for relatively hydrophobic small molecule drugs. You can use solvents like DMSO, which will basically allow small molecules to dissolve through the skin. You can't use DMSO for proteins or peptides. So the approach they're using is essentially microneedles. So you put the proteins into a standard matrix, which just has the additives to help stabilize. But the patch has these microneedles, and the microneedles, when you stick them onto your skin, punch tiny holes, just like a mosquito can punch a tiny hole in you. And rather than releasing essentially an anticoagulant and an inflammatory compound, which is what mosquitoes do, the protein is just released, and so you don't get this swelling or pain. But it is, with microneedles, it's much, much less painful to deliver drugs. You don't feel the needle at all. This is an example of 3M, and they've been making microneedle patches, and they look like tiny pyramids, but these are hollow pyramids that allow a drug to flow through the pyramid and into the tip and into the bloodstream. This is another example of one where it's sort of a punch applicator, and there are now similar kinds of microneedle systems to also collect blood. So you can deliver drugs, you can collect blood through microneedle approaches. But in this case, it's trying to deliver drugs through these microneedles, or they call them microprojections. But again, it allows the delivery of small molecules and peptides. So I've gone through a fair bit about both the formulation and delivery of protein and peptide drugs. Some of the same formulation tools that can be used for proteins and peptides are used for small molecules. And in fact, a lot of the inspiration for that is coming from the small molecule drug delivery field. We've talked about nanoparticles, we've talked about micro or nano needles. So we're certainly seeing more and more use of nanotechnology in biopharmaceuticals and protein pharmaceuticals. There's even talk about trying to use both cells and viruses as protein factories, as a way of producing things. So particularly lactobacillus bacteria, which is very stable in the human gut, could be a mechanism for delivering protein drugs. They're also looking at artificial or synthetic cells as drug delivery agents. And obviously, even things like RNA is being used as a way of getting proteins expressed, which is a characteristic of the COVID vaccine from both Pfizer and Moderna. Now, in the early days, and maybe this is only 25 years ago when people were thinking about nanotechnology, the visions they had were that you could make, nanoscale robots out of, metal and little nano computers that would go around just like robots with lasers, and they'd hunt around for viruses, look at them, and fry them with their laser. That's actually physically impossible. You're basically in the quantum realm. You can't make machines and machinable parts. But at the nano or micro level, there are cells, bacterial cells and viruses, which can be programmed and can be designed to recognize foreign things or to release payloads or to target certain areas. And in fact, more and more work has gone into the development of, if we call them smart pills, but these are bacterially engineered systems or virally engineered systems to produce proteins, or things with smart materials like nanoparticles. Another approach is to basically put a box around cells. And so techniques in nanotechnology allow you to machine metal at the micron or even submicron level. And so this is an example of an artificial islet cell where they basically create a container, a metal container, about 500 microns, 600 microns across. And the container holds islet cells. And by punching holes that are only a nanometer or two across in the container, you allow insulin, which is only about 1 or 2 nanometers in diameter, to be released. And it allows oxygen and glucose and sodium and potassium, which are essential for cells to grow, to diffuse naturally into the container. But the cells are too big, so they won't diffuse outside the container. And antibodies are too big, and they can't diffuse in the inside the container either. And the antibodies therefore can't attack these islet cells. So this essentially gives you a way of protecting islet cells, or doing an islet cell transfer or transplant, while giving the cells the opportunity to grow, to get the oxygen and sodium and glucose that they need. But it also prevents them from being attacked by antibodies or T cells or B cells, because these are just too big. They can't pass through the holes that you've made in this little artificial capsule. So nanotechnology allows people to use things like photolithography or electron beam etching to carve in very small holes into these metal plates. And these porous plates essentially allow you to have these little jail cells, if you want, that keep the islet cells in, but keep most everything out. But it also allows the insulin to leak out. And so this has been explored as, again, another way of delivering insulin, in essence, but through essentially an artificial cell or a biocapsule. And this is an electron micrograph of the cells that are hidden behind that little capsule. And you can see kind of small holes. These are that were generated through micromachining, electron beam lithography. And it's just illustrating what antibodies can't get in, and the insulin can diffuse out. So I'm going to wrap up here and just sort of close off with a couple of summary points. So I've said this before, I'll say it again, the protein drugs are and continue to be the most rapidly growing sector in the pharmaceutical industry. It's where most of the effort in pharmaceutical development is happening. Most of the new drugs and the breakthrough drugs are happening in the areas of peptides, proteins, vaccines. I think it's also important to remember that many of the cures for the really difficult chronic diseases, like Alzheimer's disease, cancer, MS, many autoimmune diseases, will probably depend on protein-based drugs. We see that the most successful therapies for these things are either antibody-based drugs or protein drugs or enzymes. And so given that the diseases of greatest concern will probably be treated or mitigated through protein drugs, then it means that we have to find good ways of both delivering them and stabilizing them and formulating them. Now, I've highlighted just how difficult it is to work with proteins. They're big molecules, they're not very stable. We also have identified that, yes, most proteins can be delivered by injection, but if there are better ways, we'd love to find them. And certainly the microneedle injection processes seem to be among the most promising. There are examples of oral delivery of peptides and some protein drugs. There's a lot of work to try and use smart materials, and people in the field of both materials engineering and chemical engineering and pharmaceutics are working on coming up with better materials all the time. I think maybe it's a little optimistic. Maybe in 3 to 4 years' time, maybe 5 to 10 years' time, we'll start seeing more drugs having some oral formulations. I'm more optimistic actually with the microneedle delivery systems as being able to deliver proteins in a painless and simple, consistent manner. So with that, I think that covers today's lecture. If people have questions or in the chat, so I guess there's one question from Adam about how do you get the small boxes into the pancreas? In most cases with these metal biocapsules, they don't have to be in the pancreas. They could be just stitched in under the skin. So it becomes an artificial pancreas. All it needs is something that can be sensitive or can respond to blood glucose. And that's why you want to have glucose being able to permeate. So it could go under your arm or under your foot. So it doesn't have to be a major surgery where you're actually putting something into the pancreas. Were there any other questions? Yeah, so biocapsules are made out of titanium. So titanium is biologically inert. This is why a lot of the artificial joints and knees and hips are often made out of titanium. So the biocapsule is also made out of titanium. So if there are no more questions, or if people don't have any more questions, then I guess we can wrap it up. I'll stay on for another minute or two in case people have some last minute question, but I think we can call it a day. Thank you, everyone. Okay, bye for now.